welcome everybody to this session. So we're going to be focused on dementia, and that's all the way from early stage drug discovery and new treatment discovery through to dementia care. And we've got a great panel who represent different stages of that process. I'll get them to introduce themselves. My name's Dave Sharp. I'm a neurologist. I've been at Imperial for about 25 years now, and I lead one of the centres within the Dementia Research Institute. So my centre is called the Care Research and Technology Centre, and we're focused at the care and technology end of the spectrum, if you like. But the DRI has a whole range of different research activity going on, from very basic drug discovery work all the way through to care work. So I'll pass on, and we'll just go through the panel and introduce themselves.
Hello everybody, I'm David Reynolds. My day job is running a biotech called Locus 23 Therapeutics, which is focused on small molecule treatments for Huntington's disease and other triplet repeat diseases. But I've got kind of various hats. I also helped to found another biotech working on Alzheimer's, primarily called Astronautics. And as it says up there, I'm a venture partner, so a scientific advisor to the Dementia Discovery Fund, which is a venture capital fund managed by SV Health Investors here in London, and the DDF, the Dementia Discovery Fund, was set up specifically to invest in dementia, because this was an area that didn't get enough investment in terms of bringing new drugs through to the market. And before that, I spent 20 odd years in large pharma.
Thanks, David. Robert?
Hello. I'm Robert Delay. I'm a neurologist, and for the last 25 years I've worked in industry, in GSK and ASI, and now in GSK again, leading various large projects on developing the disease modification drugs for Alzheimer's disease, including lecanemab. And I'm still working on another big project that we've got, so I try to bring the industry perspective on drug development to the panel discussion today.
Katharine?
Hi everyone, I'm Katharine Willmette, and I'm the Director of Adult Social Care in Hammersmith and Fulham Council. And what that means is I'm responsible for strategy, delivery, planning and operational functioning of adult social care in Hammersmith and Fulham. I also work with our colleagues in the voluntary sector and with our health colleagues around supporting people with dementia.
Francesco?
Yeah, hi everyone, my name is Francesco Aprile. I'm a senior lecturer here in the Department of Chemistry. I'm also group leader at UKRI FLF, and our team work at the intersection between biophysics, chemistry, and biology. So we like to investigate disease mechanism and potentially develop molecules to understand the disease, but also potentially to try to translate them into diagnostic therapeutics.
Great. So we've got a few initial questions that we'll discuss, but then there's going to be plenty of chance for people to ask questions. So please think of questions and then get ready to contribute, if that's okay. So we're just going to start broadly with the panel's comments about new directions for diagnosis and treatment in dementia. So it's been a pretty, I think, exciting time over the last 2 or 3 years, where we've gone from a situation where having a whole series of failed drugs over many years to a situation where there's really quite a lot of excitement about the opportunities at the moment, and also the new developments in diagnostic biomarkers, I think, are changing the landscape of how we might identify people at early risk. So I'd just be interested in the panel's views on where the most exciting directions are, how you feel about things at the moment. Maybe starting with you, David.
Okay, so yes, I think it is an exciting time in dementia research. It's funny, when I look back and first joined the industry in 1997, the cloning of the amyloid genes and the presenilin genes was all very exciting, and we were going to get new therapies for Alzheimer's within the next 5 years. And that didn't really happen. But it has happened now. There are new amyloid-lowering therapies that have been approved for use, and I think that's a really exciting step forward. Now, are these the best drugs in the world? Are they the easiest to use? No, I don't think so, but they are a really big step forward in starting to be able to have an impact on diseases like Alzheimer's and slowing down that progression. And that's something that I think, certainly through my career, is something that's been aspirational, and now we are there. Now, I think there's a lot we can do moving forwards, and I'm excited by what else is in the pipeline, drugs targeting tau, looking at neuroinflammation. I think there's a whole wealth of very well thought through hypotheses being tested in different ways that I think will make a big difference over the next, say, 5 years or so.
And just, isn't it interesting your perspective on the last 20 years of you, having lived through it? What was the reason why it's succeeding now, do you think? Is there a reason, or is it just time or new approaches? What's changed?
I think there's various schools of thought. So a very basic one in terms of large pharma thinking is number of shots on goal. The more things you take into research and then into clinical development, the higher probability you have of success. Now, the key requirement for that, though, is knowing where the goal is to know if the shot is any good or not. And I think the science has certainly matured a lot in the last 20 years, as has our ability to identify patients, because being able to identify Alzheimer's patients or other neurodegenerative disease patients early in their disease course, as I think you probably all know, there's a very long kind of silent phase where the person is ostensibly healthy, even though if you do an MRI or various other biomarker tests, we can see that there is neurodegeneration ongoing. And our ability to find those people earlier and have thought through what that process is and understand it, and then apply new potential therapeutics in clinical trials, I think is a lot more advanced than it was back, when I was a lad.
And there was a bit of a caveat in your statement about the new drugs. Maybe say a bit more of that. Do you think we'll be using the new drugs widely or not?
So I think that is a challenge and one for other panel members to talk about, I think, in more detail. But the amyloid-lowering therapies, yes, they lower amyloid, but they have a certain number of safety risks, primarily amyloid-related imaging abnormalities, or ARIA, which you have to do regular safety scanning using MRI. That's not trivial, it's not straightforward to scale that to the number of patients. So the ability for us to kind of implement those drugs in standard clinical practice, I think, is challenging. Now, there's one good thing is that when there is a challenge at hand where there's a treatment, then people really look for ways of how do we sort this out and improve this. And as you go earlier in the disease course, those safety risks are lower. So now we need to be looking at patients earlier and earlier, where I think they will have A, more impact, and B, more readily usable.
Great. Robert, can you give us a sort of farmer perspective on all of this?
I think clearly over the last 25 years there have been great advances. The targets have been known for some time before we actually had the drugs. But I think one of the main things that advanced the development of the medicines is the development of all these diagnostic tools. Initially, of course, we had amyloid PET, the PIP, and then we had all these various tau traces. Now they become established, proved diagnostic instruments. And more recently, the use of the plasma amyloid ratios and plasma p-tau tests have really advanced things a lot. And I think I think a lot of the old problems, the old studies where they didn't work very well, was that there was no objective diagnostic, sort of molecular diagnostic criteria, because the tools were not available. So some of the patients probably didn't actually have the amyloid pathology of Alzheimer's disease, and the clinical criteria could not really narrow that down. And now we have those instruments, so we actually know that the patients we're treating in the trials actually have the target, have the molecular target. And therefore, of course, if they didn't have the target, it wouldn't work. So I think that's one of the big advances that allow us to not just diagnose accurately, but also now to track target engagement and to track the amount of the target reduction, target engagement that is needed in order to relate that with clinical benefits. And the cognitive instruments, well, they've been around for some time, and there's a sort of variability and uncertainty in terms of the efficacy that one sees, and they're not very huge effects. But the development of all these diagnostic tests that can also be used as the target engagement biomarkers actually help to provide objective evidence to convince the agencies, convince the regulators, and hopefully the payers too, that it's actually changing the disease pathology. And so I think if some of these drugs actually just show slowing of clinical decline, may not be so convincing. But I think these development of all these molecular diagnostic instruments really help to push the field forward in convincing people that the drugs do work and identifying the correct patients.
And on the identification of correct patients, where do you see the blood biomarker kind of work going? Is that a big change in terms of the drug development, do you think?
I think it's a big change because when I worked on lecanemab, we had to rely on amyloid PET, which is very slow and expensive, and plus radiation exposure. And we had a lot of patients who actually had the cognitive impairment, but actually did not have the amyloid pathology. So they were either just elderly, they had natural decline, aging, or they had something else. And it was a very high screen failure rate, and it was actually very expensive, very cumbersome to push people through, with most people actually not having the amyloid PET. Now, of course, it's quite extensively deployed. I mean, some of my own projects and a lot of other sponsors use the plasma tests in the clinical trials as a sort of triage step. Okay, they're not 100% accurate, but I think you can certainly screen out a lot of patients who are less likely to have amyloid pathology, and then it moves towards a confirmatory test. I think increasingly, once these plasma tests are approved by agencies, they may just replace the imaging tests. I think at the moment we still use imaging CSF and imaging tests for definitive diagnosis. Partly for some agencies that's what they prefer, but hopefully that we won't have to do that step in the future. But it certainly cuts down a lot, speeds up a lot of all these patient recruitment and screening processes. Now, I think in the US, I think it's increasingly more used in ordinary clinical practice for diagnosis, and I think some of the guidelines say that if patients are thinking about taking anti-amyloid drugs, then they probably need something confirmatory. It's not being extensively used in other regions, but I think it will probably come that way. I think it's not so easy to deploy them in Europe.
And both the biomarker tests and the drugs are most effective, obviously, for amyloid treatment. So if you think about the kind of tau element of it, where are we going with that? So are you optimistic about having tau agents that work, or the ability to screen accurately for tau?
So in terms of screening, there's some of the plasma tau tests are reasonably good. p-tau217, I think that seems to be emerging from the conferences as one of the front runners for looking at not just the amyloid, but also to some extent the correlation with tau pathology, the accumulation of tau pathology. It doesn't correlate so well with tau pathology once you get to the more advanced stages, but it can. But I think if you're trying to catch people with early stage of tau pathology, which is the stage where we want to try and treat, I think it probably will be useful in identifying those patients who can then be further characterized by the standard tau PET, because some of these have been approved. I think in terms of tau as a target, it's more challenging, and partly because the most of the drugs now, still the antibodies that have great difficulty getting to the sites where the tau is, inside the cells or at these sort of synapses, so they're not so accessible. People are still trying to work out which epitope the antibodies really should attack in order to have better effects. So some of the second generation ones actually hone in more on the microtubule binding domain, which is where the aggregation processes occur. So those may be more promising, and of course ASI is leading one of those studies. I think other companies are coming along. So I think I'm hopeful, particularly seeing some of the anti-tau antisense oligonucleotides that actually reduce the tau, and it seems to be able to reverse some of the tau pathology. I suppose as long as it's not too advanced, you may be able to reverse it to some extent. So we're waiting to see whether that will have some benefit in slowing the clinical progression, some of the Biogen studies. So I'm hopeful, but I think tau is, the antibodies, the drug itself may not be so straightforward. Yeah, just to get inside the cells.
Perhaps go to Francesco next. Do you want to comment?
Yeah, sure. So I think that what excites me the most about the field in this period are probably two areas. So as my colleagues have said, now we have a better, let's say, diagnostic potential. Like there are several technologies that have been developed, and these probably inform better research as well. There is an understanding now that besides the contribution of inflammation versus aggregation in the development of dementia, and also very interesting aspect that were discussed that several times we have pathology in the brain but no symptoms, or the other way around as well. It's also the fact that now there are many more gray areas, like many cases we can have Alzheimer's disease patients which also display other type of aggregates. Traditionally, we used to think about tau and the beta, but now we can also find alpha-synuclein, which is associated predominantly with Parkinson's disease. And so it seems that, we have a growing cases of mixed dementia that perhaps have always been there, but we didn't have the technology to actually document these diseases. And so now these open new frontiers and new possibilities also for basic research. We can actually try to understand how these proteins interact in the disease context, and how, for example, inflammation or an inflammation background in the brain can contribute. So this, in my opinion, is a very growing area, and that offers several opportunities. And then second is the use of biologics like antibodies as a treatment, that again, clearly very recently we had several cases that show some promise, like aducanumab, lecanemab, then some were withdrawn by the companies, but still these show that perhaps that's a direction that could benefit. On the other hand, these also show that we need to improve administration techniques, also for example, to minimize side effects like ARIA, like this inflammation in the brain that is unwanted. And so it's just to say, two big areas like improvement of diagnostics, and then a new way to see diseases and dementia, like redefining a little bit what we call Alzheimer's, Parkinson, or dementia with Lewy bodies and so on. And on the other hand, the development of biologics that anyway trigger actually other questions, like how can we get more of these biologics into the brain without inducing damage. And so, I think that, exciting moment, mainly triggered by the fact that I see in the UK there is a strong collaborative network, also, for example, boosted by the Dementia Research Institute and several other institutes that are working together towards specific aspects, and the groups working together towards the same problem, approaching it via the biophysics side, the chemistry side, the biology, and so on. So yes, I think that this is probably good. There is momentum, and so I think that we're advancing faster than before.
Okay, so we'll come back to the collaborative question in a bit and discuss that. But Katharine, it's obviously really important that we think about how we care for people who are affected by dementia now. We've got some major challenges about how we do that in a way that's cost effective, I suppose. Would you like to give us your thoughts on that?
Yeah, sure. So we've got the latest data we have is that around 6.1% of the population in Hammersmith and Fulham have dementia. So it's a significant number. It's about 1,500 people. That goes back a couple of years, so it's probably higher than that now. I'm really interested in the developments around early diagnosis and treatment for lots of reasons, but not least because I think it's helped to drive a real cultural change in how we view older people, and particularly older people with dementia. And what that's meant is that there is much more of a focus on people and their needs as individuals, as opposed to, well, dementia is just a part of ageing, which is certainly how it was viewed 20, 30 years ago. And so we're trying to be very, very responsive to people and very responsive to people's needs. And the question that springs to mind when we're talking particularly about early diagnosis, but also in the development of treatments, is what does that mean for the individual and for their family? What do we need to put in place that we don't do at the moment? Because people are being diagnosed more quickly or earlier in the process, in the disease process. What do we need to do differently, both as a care provider, but also as a collaborative partner working with health sciences and health services and working with voluntary sector partners, because that landscape will change hugely over the next few years. And it's really heartening to hear colleagues talking about the advances that are being made and the rapidity of those advances, but we mustn't lose sight as well of the individual at the centre of that and what they need to help them on that journey as well. And some of the conversations around question marks around you deliver some of the treatments, how you deliver some of the diagnosis, or how you work through the process of diagnosis is really important, and that applies equally to care as well. We've got to keep up on the care service side with the health advances, with the scientific advances around dementia. So for me, it's really exciting that those opportunities are coming forward, but it just means we have to think a little bit differently about how we work together to provide support for individuals and families because of the new scientific advances.
Great, thanks very much. So maybe just switching from the opportunities to the challenges briefly. Maybe you could kind of each highlight what you think are the biggest challenges and how we might think about addressing them.
Maybe I could come back to a point that was kind of raised earlier, that we now have different modalities, different types of drugs. So if you look at the vast majority of drugs that are in use for treating CNS disorders, they're small molecules. And the reason is that those can get into the brain generally reasonably easily, and therefore, to Robert's point, you can get target engagement. It actually hits the receptor, the enzyme, whatever the drug target is. Now, antibodies get into the brain a little bit, enough that we can see effects, but only a very small proportion of the antibody burden that's dosed to a patient actually ends up in the brain binding to amyloid. But the advances in the way in which we can bring forward new modalities, I think, over the last 20 or so years is making an enormous difference. So now there are ways of helping to shuttle antibodies into the brain, or increasingly there are gene therapies which can be packaged in AAVs that are tropic for neurons as opposed to all ending up in the liver. There are various ways of targeting RNA. So now we have the kind of full gamut of choice from the gene you're trying to target, the RNA it produces, or the protein. And different modalities have different benefits and downsides. So I think that gives us a lot more tools in the armamentarium. The other thing is about advancing understanding of how the biology of these target proteins work. The amyloid antibodies that are approved were not the first ones that went into clinical trials. Absolutely agree with Robert that some of those trials may have failed because a third of the patients in the study didn't actually have Alzheimer's disease. But also we've learned that there are different areas of amyloid or tau or alpha-synuclein that you want to target for maximum effect. And I think that improving precision really helps us with the new tools to kind of try and pick our way through what is a very kind of complex disease, or set of diseases when you consider neurodegeneration more broadly, to try and kind of move forwards.
Okay, thanks. Robert, challenges?
Well, I was saying that I could look at it at different stages of drug development. I think in the early sort of drug discovery stage, there are lots of targets in the molecular targets, and apart from the obvious amyloid and tau, and it's not entirely sure which ones of these actually beyond amyloid and tau people should focus on, and they're likely to have some effect on slowing progression. And a lot of targets now focusing on microglia, inflammatory reactions, which look pretty attractive and reasonable, and some of these have a genetic basis to them. But how well which targets are likely to work, there's a lot of redundancy in some of these systems. So which ones are actually likely to have an effect? We don't have a straightforward way of identifying or of determining that. We could try some of these animal models, and they're not highly predictive. So often you might just have to, various sponsors will have to try many different targets and eventually find some that can move forward, a lot of them actually fail. So that's part of the early stage. In the mid stage of development, I think even if we do have new targets, is determining how much level of exposure and target engagement, target reduction is needed. So it's actually taken at least a decade or so to work out that for the amyloid clearance, you need at least 80% or something that maybe completely clear the amyloid to have a good clinical slowing, and it's not a very huge slowing. And some of the earlier generation anti-amyloid drugs, and some of the more recent ones actually didn't reduce amyloid sufficiently, and so they didn't work that well. So, but for new targets, it's really very difficult to know how much you want to go for. And so that's the other challenge. And at the late stage development, the one challenge I see is really how a product that has already been shown to work, it may actually be a new target as well. how it differentiates from the approved treatments. Because it's competitive, and so some of the earlier drugs tend to get used first because people are just more familiar with them. And how should a new one be positioned therapeutically, particularly when it's not always easy on clinical endpoints to differentiate lecanemab from some other new mechanism, even if that new mechanism actually works, and how it actually fits into the hierarchy of treatments. That's something we don't know. And it's not so easy to sort of work it out from the trials, right? Often we need head-to-head trials. And I think we have lots of treatments for migraines, but don't think really there has been any extensive head-to-head comparisons. And it's all just based on indirect comparisons with trials.
Thanks. Katharine?
I think from a local authority perspective and from a social care perspective, it's very much about investment. And how do organisations that are short of money and have limited opportunities to invest in different areas of support and care, how do they make the decision about what we're going to do differently and where we're going to invest? Because I think the research is probably way ahead of our thinking on that, and we haven't started to think about how do we need to organise ourselves differently, how do we need to work with health services differently so that actually we can respond to these treatments and the scientific advances. I think the biggest hurdle, probably in the short term anyway, is money, and actually how do we spend our money wisely to get the most impact out of what's being developed. Because if we don't see an improved outcome, improvements in people's well-being, improvements in people's support, it's very difficult then to focus on that investment.
And you mentioned about the kind of the connection with the health service. I mean, that kind of social care health connection, which has historically obviously been a challenge. Do you think we're moving in the right direction? Are we getting more integrated, or is there more to
I think we're moving in the right direction. And in Hammersmith and Fulham, certainly, we're doing work specifically around supporting people with dementia, or we're starting to do work, and we're trying to do that together. I think there are all sorts of huge organisational barriers that sometimes get in the way, but absolutely that is the right direction, and it's the direction we're moving in.
Francesco, challenges?
As a researcher, I would say that the complexity of the disease, which I think it emerged from this discussion, the fact that the biomarkers are very complex. When we say amyloid beta or tau, we're talking about proteins, molecules that can acquire different shapes, and chemically they will be the same, but different shapes may have different toxicity, and then this is combined again with inflammation, so very different mechanism. So that still is a challenge, even if we are progressing in understanding the disease and in targeting some of these biomarkers, require quite a substantial research effort. And then, yes, I agree that for many years there was this misconception that dementia was a part of normal aging, and therefore we should always bear in mind that is actually not. And so keep it as a national priority, and focusing investments for research and care, I think is important, and perhaps these a few years ago have limited a little bit both this area. So now it seems that there is momentum, we should keep investing.
Okay, so I just have another couple of areas maybe we could go through relatively briefly, but and then open it out if that's all right. The first one is around caring for dementia, and it's Katharine, maybe you can sort of comment on that. Where do you think we're going? Where should we be aiming, I think, in thinking about social care and personalising care, as you've mentioned?
I think I've talked a little bit about that in terms of how we need to keep up with those scientific advances, and we're trying to do that. We have relationships in Hammersmith and Fulham, as you know, with Imperial and with a whole kind of digital programme. It's really important that we do that so that we know where things are going. But we also have to listen to what people say to us. So we have in Hammersmith and Fulham, as in many councils, but we're particularly good at this in Hammersmith and Fulham, we have really good relationships with people who have dementia and with people who support people with dementia. The Dementia Alliance being an example, the Dementia Academy, which is very much about getting people with dementia involved in research. I think if we don't do that, then we're not going to be able to anticipate what we need to do differently. And it's a changing picture all the time, and I think that's been demonstrated by what others on the panel have been talking about, very much about actually what difference does it make on the ground. I think that's really important, that's what we hang on to. And I think those relationships with voluntary organisations, with people who have dementia and people who care for dementia are at the cornerstone of that.
Great, and just maybe as a last thing about collaboration and partnership, I mean, I think in the UK we've got world-leading research in dementia in a number of areas, really, and we can, I don't think would be disputed nevertheless, necessarily, but at the same time, we've seen a kind of drop-off in the amount of clinical trial activity that's gone on in the UK over recent years, and in many ways, the system end-to-end is not functioning, very smoothly. So I wonder what people think about how we can work collaboratively or more in partnership to accelerate the progress. I mean, anybody want to take that on?
Maybe I could start just thinking. We have all the parts in this country, absolutely, but it's making sure they're linked together and work efficiently that I think will have the biggest impact. So you've just mentioned that clinical trials in the UK have been declining over a number of years. So what's the reason for that? So there are many elements to it, but making sure that we are able to set up and run a clinical trial efficiently and can quickly recruit patients, I think, is a key part. And I'm sure Robert can speak in more detail on this. But if we look at what the government's trying to do from the Dementia Mission point of view, it's around trying to enable an ongoing kind of trial-ready cohort. And this is something that has been done before, but trying to make sure that we have elderly people we're tracking so that we can use some of the new biomarkers, see where this is somebody who's perhaps 10 years from a formal diagnosis of Alzheimer's disease, start understanding on a semi-regular basis what their kind of biomarker trajectory is, and if we can work in some of the newer digital tools and ways of assessing cognitive and other function early, then it's much easier to then get these patients into a clinical trial. We also need to make sure that the paperwork side of things is sorted out, and I think the MHRA are trying to kind of move in that direction to help be able to get sites up and running more quickly, try and centralise some processes rather than having to go around the same cycle of paperwork in umpteen different sites around the country. So trying to link those pieces together to make it much more efficient. Now, it's much easier said than done, but I think people are trying to focus on this, perhaps even more so now that we see some drugs successfully being launched, there's more things coming through the pipeline, and actually it's great for patients to be able to be part of clinical trials. It helps drive an understanding that there are new medicines coming through. It helps to, in the general public, raise awareness that it's not an inevitable part of ageing. There are treatments there, and therefore get much greater engagement so those patients can get to people and have benefits.
Yeah. Robert?
Well, I agree with you on that. Having some of these pre-characterized patient cohorts actually very helpful. I think from experience of clinical trials, I think a lot of patients coming to the investigator sites still have screen failures because they don't actually, the investigators didn't even know what their previous or the current cognitive function was because either they were new or they haven't been tracked. And we still have screen failures arising from that. It's one of the very common reasons. So having pre-characterized cohort will certainly make it faster. Another area of collaboration I think is quite important is that a lot of the sponsors want to try and move the studies in Alzheimer's and dementia to an earlier stage. And I think, of course, that's a good thing. Hopefully we can sort of slow it down at the earlier stage, and it seems to work better if we keep people earlier. But I've always thought that we probably need some sort of public screening program, or maybe where there's collaboration with social care, where people are checked before they actually notice really significant symptoms, and they can be checked out. I mean, some patients are more aware than others, but if there's some kind of screening program, like what we do for cancer screening, then maybe we can pre-characterize those patients. They don't need specific treatments. They may not need a lot of support. They may be able to help with all these sort of early stage Alzheimer's trials, right? Sometimes we call them prevention trials, early stages, perhaps more size. And those patients are often quite not so straightforward to find. They usually, I think in some of the more recent studies, they're collected by these academic research centers who've got special interest. And, but trying to to find large numbers of these people in the community, and you need large numbers of them before they get to the demented stage to do the studies, because the decline is slow. It's actually quite difficult.
Yeah, I mean, I think from my perspective, I think the tools available, particularly the blood biomarker testing, is moving it to becoming much more feasible. I don't think it was ever feasible to do imaging, with PET on very large numbers, but it certainly is with blood testing. I think it becomes feasible in all sorts of ways. So that hopefully will open the door to having, a much larger number of people who are ready to go into trials. And I agree with you about the kind of the almost like a cultural shift in people thinking about the fact that there are things that they can engage in, and we need to kind of have a conversation at the national level really about that, I think. Good. So that was a very good discussion. Thank you. Maybe we can open it out to the floor. Lots of questions.
Just a comment really, and that is that Our Future Health was launched with great success last year, and a lot of people have lent into that, and it seems to be very cardiometabolic focused initially, so you can pop along to Boots or something and they'll measure your blood pressure and your blood sugar, etc., etc. But it seems to be that and the willingness now of folks to lean in to with their health data studies is a great opportunity. Our Future Health has got to be some infrastructure that seems to be ready-made for what you were just talking about, and it seems that it should be an opportunity for people who are interested in understanding how their cognitive health will change over time to join up with that, because I think a lot of the other infrastructure is already there.
No, I completely agree, and we. In many of this, we do very well in this country. I mean, Biobank is an unqualified international success. It's being used by researchers all over the world. And so we've, that's been an amazing success, right? There's no reason why, we can't build on that.
I think it's nice to get to living people before they start giving you their brain slices after death. It's, it's about having them take part already.
Yeah, Biobank is on living people, but
yes,
but I agree about Our Future Health.
Can I just touch on primary prevention, because none of us have mentioned that, and I think the cardio-metabolic kind of health piece really kind of leans into that. When I was at Alzheimer's Research UK, one of the things we used to tell people is that what's good for your, at the strapline level, is what's good for your heart is also good for your brain. So the health and lifestyle changes you can make for cardiovascular benefit are pretty much the same, that also give benefit in terms of primary prevention for developing dementia. So they, eat healthily, fitness, don't smoke, don't drink too much, etc. Are all things that have an impact. And the Lancet reviews on dementia have shown that maybe up to about a third of dementias could be preventable or at least delayed through those kind of impacts. And that is a bigger effect size, or at least as big effect size as the marketed amyloid drugs.
So, Christoph Potempa from Brain Cures. So 10 years on for Dementia Discovery Fund, has there been any success for the dementia patients, and is the Dementia Discovery Fund investing in any early stage these days, or what's the future?
So that's one for me, definitely. So just for those in the audience to understand what the Dementia Discovery Fund is, it was a £250 million venture capital fund initiated through David Cameron's government with a focus on dementia. It was then put out by the government for tender for a professional VC house to run it, and SV Health Investors won that bid and have been managing it. So what the Dementia Discovery Fund has done starting in 2015, and I think they made their last investment in 2022, I think it was, is look for biotech companies in the UK but around the world that have an interesting scientific hypothesis of how they can treat Alzheimer's or other forms of dementia, so ALS, frontotemporal dementia, Huntington's disease, Parkinson's disease primarily, and also an actionable plan. And of course that's the important thing for any kind of small biotech, is that you've got an actionable plan of how you choose your modality, take that forward, and hopefully show it works in the clinic. So of those investments that have made, I can't remember the number off the top of my head, but there's well over a dozen of those companies that have now got things into the clinic and being tested. None of those have yet made it through to approval and to patients, but certainly it's a work in progress. And it was certainly successful enough that a second Dementia Discovery Fund has been raised to kind of then look at the next horizon of starting new investments and taking it through.
Thanks. There.
Thank you. So the question, I think, kind of relates to you, David's last discussion topic, partnership. While looking for a cure for dementia, it's admirable, and I think it's the toughest part, I think, as we all mentioned, is how to actually track the biomarkers before the late stage of development. To me, I don't know, maybe I'm very naive, I think the solution is quite easy because the NHS is in a very great position. It's a centralized government body that actually decides and has an NHS app. To me, it's like, why don't you make anybody that actually wants to book a GP session go through a screening, process, 5 minutes, track that all the way until life is over, and see what it can get from that, what kind of data, and then select, even more for groups of people that have, certain characteristics, bring them in the office. I don't know, what do you guys think of that? I don't know. Because you have a digital app, you have to go to book an NHS, GP, which is actually very unique in the world. It's not in the US, it's not in the rest of Europe. You have centralized government for NHS. You have a very great position to actually do something about it.
Yeah. Comments? Why don't we do that?
No, it's easy one.
I don't know why we don't do it, but I guess just thinking off the top of my head, people would have to want to do that. I suppose people would have to want to participate in that, and maybe they would. I don't know, but I don't think I can answer why we don't do that. But it certainly sounds like a feasible idea to me.
I think maybe I'll just comment that, because we already do that, GPs already do that to some extent with cancer screening. But we may need a sort of cultural shift where people will need to screen for about their cognitive health and change their attitudes that in the same way that they take up cancer screening. I mean, that's not extensively taken up, right? That's the people who don't want to know. But one of the difficulties is that, GPs might be able to do relatively simple tests, and they need to get the time to do that. They're already very busy. We have better instruments that won't take a lot of time, right? But the follow-up, once they have been found the biomarker abnormalities, I think that's the difficult part. That's not.
Before you go to the GP, that's the screening online at home, not on the time of the GP.
Maybe I can offer a lesson from history of how these treatments evolve. If we look at how cholesterol is now screened for in a standard way when you go for a GP's treatment, it wasn't always like that. When the cholesterol-lowering drugs first were approved and marketed, they were for people who'd had a heart attack or various major cardiovascular events, and they were used to try and reduce the recurrence. Then, once that had been proven, you stepped a stage back at people at high risk of a cardiovascular event and showed that that could reduce the number of events. And then you step further back, if you've got elevated cholesterol, then over time that reduces your risk. And in many ways, that's similar to what we see with dementia. At the moment, we wait for a diagnosis and severe, relatively severe symptoms before we go, what do we do about it? Now we're in a position to start kind of stepping further and further back. But that's got to be supported, A, by science, and B, by long-term trials that can show that for somebody who's in the pre-symptomatic stage where they've got elevated biomarkers, if you do a PET scan, you can see amyloid beta, an amyloid-lowering drug or another drug targeted at neurodegeneration can have a long-term impact. And a number of those studies have been done and shown not to have impacts on patients. And I think it's important that we make sure that something works and is safe before we want to roll it out more broadly. I absolutely agree with your principle, but it's having the evidence base to kind of get to that point. And I think maybe the cholesterol story kind of tells us how it was done in the past and how we can perhaps do it more quickly and efficiently, but we could do it in this field, in dementia.
Yeah, I think the cholesterol analogy is actually a very good one, and we should be thinking about it like that. And it is quite a radical shift in how people view brain health. In some ways, it's very. I think it's strange that that brain health isn't dealt with in exactly the same way we think about heart health. It's strange that we don't do this kind of primary prevention that we do routinely in heart health. And what we need to do is to move towards these much larger trials. I mean, the cardiologists have done these decades ago, haven't they, with very large numbers of people looking at primary prevention. And we're going to have to do that kind of work, aren't we? And then just about I think your point about the NHS is a good one. It's often held up that the NHS should be this perfect ground to do this kind of work, and we should be able to leverage the national organisation of it. I mean, it's a unique. I agree. I work in the NHS. I'm very proud to work in the NHS. It does not function very well as a large-scale research organisation. So we've got to recognise that there are many, many barriers. And we don't. We don't culturally, make use of it as it could be. I think you're quite right. We could do more, but it's not so straightforward.
We ought to pick up on Kathy's point of people wanting to do it as well. If I look at my kind of day job field of Huntington's disease, that is an autosomal dominant inherited disease. So if one of your parents has got Huntington's, you've got a 50% chance of having it yourself. There's been a diagnostic test available since the 90s, but most non-symptomatic gene carriers do not take the test because they don't want to know whether they're going to get the disease when there's nothing you can offer them. And, in Alzheimer's disease, there is an element, it's not quite as stark as that, but there is an element of that. And so it's only when you have treatments available and you can offer people things that I think, at a practical point of view, when you make a decision about your own kind of decision-making, that people decide to or not to know. As a scientist, I probably would, but, I know plenty of people who probably wouldn't.
Other questions? Yeah, about that?
Hi, China L from Global Council. There's two parts to my question, really. When we saw lecanemab and donanemab, get the negative NICE decisions, you kind of saw a kind of almost a national sense of disappointment, and NICE got beaten up quite a lot in the media for it as well. And so I just wanted to, I suppose, ask the panel, is there a kind of responsibility on us to manage expectations a little bit in this space so that I guess it's patients, individuals and their families can kind of have a better sense of when this is coming down the pipeline, because again I do feel like everyone, as there's sort of a hype kind of taken up here to almost have a fall. And the second part to that question is, how confident are you over the next coming years that those barriers to the HTA process and reimbursement can feasibly be overcome?
Yeah, it's a very good question. I mean, it's worth pointing out that they do have MHRA approval, so it's not that they can't be prescribed. It's just that it's not cost effective according to NICE. And in the way that NICE make their decisions, it would have been almost impossible for them to be deemed to be cost effective, I think, given the amount of monitoring that needs to go on with them. So, I mean, comments on that from the panel?
I think from the industry perspective, what we try to do is to look at the quality of life and the social cost of those in our studies. Not easy to do, partly because these effects, these benefits actually take quite a long time to manifest, and we don't run studies for that long, for years, in order to show that it reduces the people that get institutionalized or people that need care at home. So all these sort of additional social care costs, we can't really measure a lot of those in our studies. We try to do that with modeling, and maybe we should do more of that. But the studies usually are the short term. So the sort of cost effectiveness is therefore not so easy to characterize. We don't really know, sometimes we don't really know how these models are done by these payers. So it's difficult for us to try and provide the data to convince them. There is a lot of work that we do in the background to try and assemble the dossier, the evidence to justify the costs. Yeah.
I think that point about cost and the costs that aren't counted is really important, because actually local authorities and local systems are in a position to be able to quantify some of that. And going back to our earlier question about collaboration, that might be an area that would be really fruitful, because if you're just looking at the immediate cost, actually you're missing a huge piece of information in order to make those decisions. So I think there's scope there for us to think a little bit differently about how we could contribute to some of that decision making.
I think some of the clinical studies, because we've got all these caregiver burden scales and that kind of thing, quality of life scales, but I don't think we actually quantify costs. How much people actually need home support and going into institutions and so on. So it's something that maybe we should start doing a bit more in these big pivotal trials.
But actually, if you're interested in costs, Alzheimer's Society have a new report that's just out that we've done in West London using the WISIG data. So that starts to dive into the costs in more detail. It's definitely worth a look. Other questions? Yeah, at the front.
Hello, I'm Kirill from IQVIA, Real World Evidence. I have a questions about genetic biomarkers of the disease, like ApoE4, for example. Will testing for these biomarkers change the calculation with respect to primary prevention, some of the interventions, availability for clinical trials? Just what's your take on this?
I mean, maybe I'll comment on that. I mean, the genetic element is an important element. I suppose the way we're thinking about it in DRI is the genetic risk that you might quantify by polygenic risk scores, some kind of digital assessment that one would do, as we heard about, in terms of your cognitive level, if you like. And then there's the biomarker evidence of actually presence of pathology. And the combination of those three things should be very helpful, I think, in terms of quantifying risk. And if you think about, again, the cholesterol analogy, I mean, we calculate routinely Q-risk score, which is a combination of different measures of risk, and collectively that gives you a percentage risk of developing a cardiovascular or stroke problem over the next 10 years. I think we'll move towards a situation where we do something similar, and one of the contributing factors might be, your polygenic risk, for example, for developing Alzheimer's over a 10-year period, say.
At an earlier stage in terms of clinical trials, and Robert, I'm sure, can speak more to this, often patients with a known genetic either cause or at least large risk factor are selected for clinical trials. So the progranulin positive patients, for example, there's a lot more clinical trial opportunities for them than less well-defined causes of their disease. So that can help to the point of enriching your patient population. For me, in Huntington's disease, it's actually straightforward enough because there is a gene test and you either will or won't get Huntington's. For a lot of the other dementias, it's a lot more difficult and there's a lot more risk factors than there are causative genes. But that certainly helps us improve our both basic research and clinical precision in terms of trying to run the best trial possible to see if a therapeutic works.
I'll just say briefly that we do try and characterize people's genotypes in the clinical trials, and maybe that with the pivotal trials, we should also look at, with all these cost-effectiveness models, look at how the genotype actually factors into that. And some people may, of course, develop a bit more severe disease, progress faster, they develop it earlier, and maybe those could be specifically targeted for reimbursement, maybe more convincing for the payers.
Giovanna? Yeah.
Hi, Joanna Lally from LifeArc. Thank you for your panel opinions. I just wonder, we were thinking about this continuum, this broad spectrum of co-pathologies that is emerging. The picture is much more complex than having pure Alzheimer's disease. What do you think about AI models that are helping, looking at clinical care, clinical health history, whether we could have at some point an AI-based differential diagnosis of dementia that could help also together with the blood-based biomarkers, the genetics and the digital cognitive assessment, assess maybe a risk, even in cohorts like our Future Health prospectively, maybe building on UK Biobank and other dementia cohorts that are there. It would be interesting to hear your perspectives on that.
Yeah, definitely. I mean, that would be terrific, right? And I think we should embrace AI and its application. And I think that clearly there are plenty of data and many more that we can gather that could feed AI machine learning approach for a better diagnosis. I think actually perhaps that would be one of the solutions to the complexity of the disease, like computing all the different pathology parameters and the biomarker levels to actually, see beyond what the human eye can actually see. So definitely, I think that could be one very exciting direction that we should definitely attempt. Yeah.
In the middle.
Hi, thank you. It's Suzanne here. I'm a PhD student working on aging in the School of Public Health of Imperial. It was based on a previous question about genetic testing. What do you think about the commercial use of genetic testing in a private healthcare setting? Do you think there's scope for that in the near future for disease risk prediction? Can you foresee any potential challenges, maybe government regulation that prevents the use of commercial testing, especially with the use of AI and where that's going? Is there a fear of people to get genetically tested, considering the recent saga with 23andMe? I'm just wondering if you could talk a little bit more about that.
I think it's a really thorny issue because you can see both benefits and risks. And I'm not an expert on that by any means, but whenever you've got data that's relevant to your health, I think it's important that the person is able to interpret that data appropriately. And how well somebody understands what a risk factor means, I think is a real challenge. So I'm sure GPs hate it when people Google their disease and turn up and say, I've got this rare thing that I've spotted on Wikipedia, and I think that's just an extension of that. So I think it can be a great tool. How you apply it in a way that is kind of safe and proportional, that's way beyond my pay grade, I'm afraid.
I mean, anecdotally, it's there. I have patients coming into clinic with their 23andMe kind of readout, and they say, this is my genetic risk, and then we have to try and manage that and discuss it and try and work out whether it's relevant to what they're presenting with. But it's not that it's not already happening. I think at scale, it's like anything, as you say, it's complicated when you're giving something that is complicated you don't understand, and then it's very easy to generate an awful lot of anxiety associated with that that may not be appropriate. Sorry, just at the back first, and then in the yellow jacket.
I just had a response to the question before, because I run a company that sequences whole genomes for the clinical use for the private practice. And it's taking off at the moment. There are clinicians who are starting to move to whole genome data rather than just doing genetic panel tests. And if it's done at high quality and it's done and communicated correctly, it's the future. It needs to be used more. I think there's the main misnomers that we see quite a lot is that there's a confusion between genetics and genomics, where genetics is being paraded around as whole genome sequencing and lifelong tests, and also the amount of coverage and the genome quality that is being used. We're not quite decided on what those parameters are, but we're starting to make headway in making that happen.
Is there a cost-effective to get quality without
No, don't waste time.
So if you could. Okay, we've got a question here, I think.
Hi, it's just a thought. I mean, it seems that with the polygenic risks and the complexity of dementias, do we too often focus on people who progress and why they progress and then trying to treat them down the chain? And with all this information, should we actually flip it and look at the people that have all the high-risk genes or have a family history that don't progress? And maybe there's more answers in the people that are avoiding Alzheimer's, whether it's in what they do behaviourally. N of 1, my grandmother had dementia in her 70s. My mother was seemingly going that way, but then I got her to drink lots of water, and it's an N of 1 observational, but she seems to be cognitively a lot better. But should we study, what those people that we think should get Alzheimer's and have the plaques that don't develop symptoms?
So I can give you a real-world example of that, or at least a research-world example of that, which is funded by the Dementia Discovery Fund. So Alcamab is a UK-based biotech that was founded on looking at resilient populations. So the person who smokes 60 a day and is now 80 and still doesn't have lung cancer, or those that have a really high polygenic risk score for Alzheimer's disease and yet don't have any plaques or A beta or tau, etc. And by looking at those individuals, what they looked at was not the genetics, but actually their B-cell repertoire and what antibodies had they generated that were essentially a kind of N of 1 kind of experiment in terms of blocking their pathology. And that from that, they've identified a number of different antibodies that are present in a few individuals, and then they've kind of confirmed this in others. And then they've taken those antibodies and turned them into a therapeutic that they're now taking through to clinical trials. So yes, those who don't get disease, who are often a lot more difficult to find because they don't go to the doctor, can be very insightful in helping us understand disease and potentially treating it.
So that's probably a very good place to finish. It's been a very nice discussion. Thank you to the panel. I think there's time for networking and donuts now. Is that right? Yeah. Okay. So thanks everybody. Thanks for the questions.